In this video, we're going to be going into a bit more detail involving neurons and certain properties about them that I didn't discuss in my previous neurons video. So we'll start off with the anatomy of a neuron. So a neuron has its cell body, its cell soma, and connected to it are portions that receive information, and these sections are called dendrites. These dendrites will receive the information, and because of this information, the action potential is able to be propagated along the axon and eventually to the axon terminal, where the whole process of exocytosis and transmission of signals from neuron to neuron occurs. To start off with, we have the cell soma. Now, connected to the cell soma itself, we have dendrites. I'm going to draw dendrites in a purple color to make it really clear. So these dendrites stick out of the cell soma and they receive information. Now, there's a small portion between the cell soma of the neuron and the axon itself, and that is called the axon hillock, and that is the site of where the action potential actually starts. So I'm going to draw that. This is the axon hillock right here. We'll say this small little rectangle, and then the actual axon itself is this blue thick line. Okay, I'm going to go into a bit more detail about it after, but essentially all you need to know is that dendrites receive information. This information is transferred through the cell soma to the axon hillock. The axon hillock is the site of where the action potential starts. The action potential is propagated along the axon and eventually reaches the axon terminal. Okay, another key property about axons itself that I just want you to understand is that axons themselves can actually be very long. Now, the next question is, what signals are actually conveyed by these neurons? And it's actually a very simple answer in that the signals that are conveyed are binary on/off signals. So it's a very simple command. And then moving on, we have the lipid membrane, and interestingly enough, just to understand, neurons are actually covered in a semi-permeable lipid membrane, which is a fat membrane. Like, this helps maintain the internal environment of the neuron, controlling what can actually enter it. Permeable. Yeah, so semi-permeable membrane, and this helps just control that internal environment of the neuron. In the resting state of the neuron itself is actually polarized, and what that means is that the membrane potential is negative. It's only when the action potential process is initiated and the sodium channels open, that's when the depolarization and the rapid increase in membrane potential occurs. But prior to that, in its resting state, it actually has a very negative membrane potential. Has a negative membrane potential at resting state. And what this means is that the ion distribution in itself is quite unequal, and that's why it's called polarized. Yeah, they're called polarized. Alright, great. So that's just a little bit about the lipid membrane controlling what can enter the neuron itself and the negative membrane potential that the neuron has at its resting state. Now, moving on, we're going to be talking about a process that we have covered in a previous video, the Neurons and Action Potentials video. The action potential process is all or nothing. The ion channels will open, letting ions enter and exit. This is in the processes of depolarization, when the sodium channels will open, allowing the influx of sodium, and the repolarization stage, where the potassium channels will open and allowing potassium to leave the cell. It really depends on the stage of the action potential and what is actually occurring within the neuron. The two general states that I want you to really understand is that it's either off, it's polarized at rest, where it has an unequal ion distribution, or it's on when it flips into its depolarized stage, and this is when it's positively charged, and it's exemplified by the influx of sodium. So that's when the membrane potential becomes very depolarized. And this is what I was talking about before as well, about the neurons sending binary signals, either being off or on, right here. What that means is it either triggers the membrane to stay polarized at its resting state, or trigger the action potential process. The neuron is actually only depolarized for a few milliseconds, but it can spike. It can switch on and off very quickly, many times per second. Okay, let's keep talking about this action potential, because there's a few other points that I wanted to go over. So, it's localized to a small segment of the membrane and spreads along the membrane itself. So it starts off, this is the key thing that I didn't really mention in my previous video. The action potential actually starts in a region called the axon hillock. This axon hillock is located between the cell soma of the neuron and the axon itself. So it begins in this axon hillock, spreads through to the axon, spreads across, it propagates along the axon, and then to the axon terminal. So in terms of specifically the direction of where it begins to where it ends, the action potential begins in this axon helix, moves through to the axon, propagates along the axon, and then eventually arrives at the axon terminal where it triggers the exocytosis of vesicles containing neurotransmitters. Another thing is, in the previous video, I mentioned how the influx of sodium in an action potential causes the depolarization phase, and then the opening of potassium channels causing them to leave causes the repolarization phase. But the stage at which the membrane potential becomes more negative than it was at its resting state, the hyperpolarization stage, its purpose is to prevent the action potential from coming back. It essentially keeps this a one-time process where the action potential is complete and it doesn't keep going in between, in and out within the neuron itself. The next thing we're going to talk about is myelin, and I did actually talk about this a little bit in my previous video, but I just want to make a few points a bit more clear. So yeah, myelin is essentially a fat structure within the neuron itself. It speeds up nerve conduction, allowing action potentials to jump across myelinated sections to nodes of Ranvier. And it jumps along each of these gaps and essentially allows the action potential process to occur a lot quicker. Imagine that this is an axon, this black rectangle that I've drawn here. So let's say that this portion here, this red portion that I've drawn here, is a myelinated section. And this is another myelinated section right here. So the action potential, which I'm drawing in the blue color, will jump across these myelinated sections to these white portions. And these white portions are, say, just for the sake of an example, they're white, but they're called nodes of Ranvier. So they'll jump across each of these sections, across the myelinated sections. Now, what that does is the length of myelin is the perfect length to allow the neuron to continually initiate the action potential at each of these little gap sections. And it just speeds up the nerve conduction, speeds up the action potential process as a whole. An example of this that I wanted to go over that I recently learned, which I thought was really interesting, is the two types of pain fibers. There's one myelinated and one not. The first one is the pain fiber A delta, so pain A delta fiber, which is located in the peripheral nervous system, and this is myelinated. And the other fiber is the C fiber, the unmyelinated pain fiber. Now, the interesting thing is when you first, say, for example, if you touch a hot stove, the first pain fiber is the A delta fiber. It's the first one. Because it's myelinated, you will feel that reflex to pull your hand back quickly. And then the C fiber, the unmyelinated pain fiber, is the pain that you feel after you pull your hand away, maybe for a second, and then that pain kind of sinks in again. That's the C delta. Because it's unmyelinated, it takes longer for that signal to propagate. A delta, which is myelinated. And I'm using the example of touching a hot stove. So A delta myelinated causes initial pain that makes you pull your hand back in a reflex motion. And the C fiber, the unmyelinated C fiber, this is the pain that sets in maybe after a second. If you didn't have an A delta fiber and you only had this C fiber, this unmyelinated pain fiber, your hand would likely stay on the stove for maybe a second longer, and that could actually cause even more tissue damage. So that could possibly be even more dangerous depending on the situation. That's just something about myelin and how the action potentials will jump across these gap sections across the myelin, over the myelin. And a little example that just helps you understand how myelin speeds up the action potential nerve conduction process. Now, we're going to be talking about a really interesting process of neurotransmission and the process of synapses. Neurons will form small junctions, small gaps between the axon terminal and the postsynaptic membrane called synapses. These neurons will communicate across these synapses. Okay, so I'm going to explain the whole process in one go, and then following that, I'll break down each individual step and make it more clear. So yeah, just a few things for your own knowledge. The neurons will form small junctions called synapses, and this is the region between the axon terminal of one neuron and the postsynaptic membrane of another neuron, and neurons themselves will communicate across these synapses. The action potential will begin in the axon helix. And remember, that's just the region between the cell soma and the axon, which is this yellow region that I've drawn in this drawing here. So this is the axon helix. It's between this blue thick line, which is the axon, and the cell soma here. So the action potential begins in the axon helix. The signal is propagated along the axon. Now that the signal is propagated along the axon, the signal will eventually reach the axon terminal. As the signal reaches the axon terminal, the influx of calcium ions will occur, and this will cause exocytosis of vesicles containing neurotransmitters, and these vesicles will bind with the membrane of the neuron on the axon terminal and release the stored neurotransmitters out into the synapse, which is the gap between the axon terminal and postsynaptic membrane. Okay, so signal reaches and now when the neurotransmitters are deposited out into the synapse via this process of exocytosis, there are actually large proteins on the postsynaptic neuron, so that's the other neuron, that function as receptors, and these neurotransmitters will bind to their complementary receptor. It will not bind if it's not complementary, but if it is complementary, the neurotransmitters will bind to the receptors on the postsynaptic neuron, and this binding will either trigger an excitatory response or an inhibitory response. For example, if the neurotransmitter GABA binds to a receptor on the postsynaptic neuron, GABA is an inhibitory neuron, which causes the influx of chlorine ions, which will actually stop the action potential process. Chlorine will further polarize the neuron, which will actually cause the membrane potential to decrease rather than to increase. So it'll actually, it'll inhibit that action potential process. Neurotransmitters bind. Potentially, they don't have to always cause this opening of an ion channel. Now, just to give you an example as well, this excitatory response could be the neurotransmitter glutamate binding to a complementary receptor on the postsynaptic neuron. And what this would cause is it would cause the ion channel on the postsynaptic neuron to actually open, and the influx of sodium to again cause the depolarization action potential process. So the glutamate is an example of an excitatory neurotransmitter. Okay. That may seem like a lot of information because, well, it is a lot of information, but I'm going to actually draw that for you now just to really help you understand this process and solidify that knowledge. So let's quickly break this down. Dendrites will receive information, the information will be transferred through the cell soma, and the action potential will begin in the axon helix, this little yellow portion here. The action potential will be propagated along this blue thick line along the axon, where the processes of depolarization and repolarization will occur, and hyperpolarization as well. And as the signal reaches these green rectangles here, influx of calcium will occur. And this influx of calcium ions, as you can see here, these little black circles are vesicles, and the small dots that I've drawn inside them are neurotransmitters. These vesicles will bind to the membrane on the axon terminal, and they'll release the content within them. They'll release the neurotransmitters out into this gap here, which is the synapse. Make sure you understand. So this is the synapse, this gap. And this orange portion is the postsynaptic membrane. So these small neurotransmitters, these dots, they'll bind to their complementary receptors on the postsynaptic neuron. Now, as they bind to these postsynaptic receptors, this ion channel will open, and what that would cause, it could potentially cause an influx of sodium, which would trigger another action potential. It could cause an influx of an influx of chlorine, which could cause a more polarized membrane potential, which would inhibit the action potential process. It really depends on the neurotransmitter that binds to the postsynaptic membrane. It could either cause an inhibitory or excitatory response. Neurotransmitters will be released out into this gap, this synapse, and this is where they will bind to the postsynaptic membrane of another neuron and either trigger an action potential or inhibit an action potential process. If neurotransmitters actually stay in this synapse, this gap, for too long, they will be likely destroyed by enzymes, or the presynaptic terminal, this green portion here, will actually suck up the neurotransmitters again in a process called reuptake. A lot of drugs actually target this reuptake process. They prevent the presynaptic terminal from actually reuptaking the neurotransmitters, which will keep the neurotransmitters in the synapse, prolonging their effect. So this is particularly effective for certain psychological conditions. Okay, that is the neurons part 2 video. That is everything I wanted to cover in this video. was essentially just going over a few processes and a few particular things that I didn't actually mention in my Neurons and Action Potentials video. Hopefully this video helped you understand some of these processes in a bit more detail. It definitely helped me solidify my knowledge of these recently learned topics. With all that being said, that's all we're going to be covering in this video.